The third judge doesn't use the negative binomial scoring system at all. She transforms the wines into a different scale, applies a completely different set of criteria, and arrives at remarkably similar conclusions, often faster than the other two. Limma was originally designed for microarrays, continuous data modeled with linear models. RNA-seq counts are discrete. Voom bridges the gap, transform counts to log counts per million, then estimate the mean-variance relationship. The Voom function computes precision weights for each observation. High-variance observations get low weights. After Voom transformation and weighting, the data behaves enough like continuous data that Limma's linear model machinery applies. It's not a count model. It's a weighted linear model applied to transformed counts. Why does this work? RNA-seq counts have a specific mean-variance relationship. Variance increases with the mean. Voom models this relationship as a smooth curve and assigns weights accordingly. Low-count genes, high variance, get low weights. High-count genes, more reliable, get high weights. After weighting, the residuals are approximately homeostastic, constant variance, which is what linear models assume. The Voom weights effectively convert the count problem into a weighted regression problem, and Limma's empirical moderated t-statistics handle the rest. Limma's advantage is flexibility and speed. The contrast matrix specification handles any experimental design, pairs comparisons, factorial designs, time series, dose response. The DREAM extension adds random effects for repeated measures designs, patients with multiple biopsies. Moderated t-statistics borrow information across genes to stabilize variance estimates, the same empirical base principle as Dussock's dispersion shrinkage. And Limma is fast. For a dataset with 20,000 genes and 50 samples, limma-voom finishes in seconds. Dussock takes minutes. Edger is in between. Single-cell RNA-seq adds a complication. What counts as a replicate? A cell is not a replicate. Cells from the same sample are not independent. The replicate is the biological sample, the patient, the mouse, the culture well. Pseudobulk analysis aggregates all cells from each sample into one profile per cell type per sample, then applies Dussock, Edger, or limma-voom to the Pseudobulk profiles. This correctly treats samples as replicates. Cell-level tests, Wilcoxon, mass hurdle model, are useful for exploratory marker gene discovery but have inflated false positives because they treat correlated cells as independent observations. Practical consensus for bulk RNA-seq, run Dussock as the primary analysis. For validation, also run Edger and Jack overlap. Genes significant in both have higher confidence. For complex designs or large datasets, add limma-voom for speed and flexibility. For single-cell RNA-seq, use Pseudobulk aggregation with Dussock or Edger for formal differential expression and Wilcoxon or Finn markers for exploratory marker identification. Report which method you used and how many replicates per condition. 3 judges. Same wines. Dussock for simplicity and trunk fold changes. Edger for flexibility and complex designs. Limma-voom for speed and the most flexible contrast specification. For scRNA-seq, Pseudobulk is the standard. Aggregate cells to samples, then test. The taste test reveals the wines, not the judges.